Hello, esteemed scholars. I am honored to share with you today our paper published in iMeta, titled, Two telomere-to-telomere pig genome assemblies and pan-genome analyses provide insights into genomic structural landscape and genetic adaptations. Since the publication of the human T2T genome, T2T genomes of several animals, such as chickens, sheep, and goats, have been released. However, the T2T genome of pigs has yet to be published. To address this scientific gap, our study successfully assembled T2T genomes of two representative Chinese pig breeds, the Min pig and the Rongchang pig, respectively. In this study, our highlights include providing T2T genomes of Min and Rongchang pig, revealing structural features of complex genomic regions like centromeres. Based on the T2T framework, we integrated public genomic data to construct a pan-genome and a graph-based pan-genome. Additionally, through multi-omics integration, we identified candidate structural variants related to temperature adaptation in pigs. Finally, we demonstrated that the graph-based pan-genome improves the resolution of genome-wide association studies and the accuracy of genomic selection. Our study is divided into 6 main parts: 1. Genome assembly, annotation, and evaluation; 2. Telomere and centromere identification; 3. Pan-genome analysis; 4. Structural variation identification and graph-based pan-genome construction; 5. Heat and cold adaptation analysis in Min and Rongchang pigs; 6. Application of graph-based pan-genome in pig breeding. We assembled the T2T genomes of Min and Rongchang pigs by integrating multiple sequencing platforms and assembly methods. The Min pig genome is gap-free, with a contig N50 of approximately 143.37 megabits and a total genome size of about 2.66 gigabits. The Rongchang pig genome contains a gap in the repetitive sequence region, with a contig N50 of about 143.96 megabits and a total genome size of around 2.68 gigabits. We also identified the centromeric regions of both genomes. The centromeres on chromosomes 1 to 12 and X are classified as metacentric or submetacentric, while those on chromosomes 13 to 18 are classified as acrocentric or telecentric. By integrating two T2T genomes and public data, we conducted a pan-genome analysis, identifying 25,472 gene families and 194,234 structural variations. The T2T genomes revealed a rise in gene family numbers, particularly variable ones. Genes with structural variations showed lower expression across tissues. We also built a graph-based pan-genome and tested genotyping accuracy with 246 high-depth sequencing samples. We conducted a heat stress experiment on Rongchang pigs and obtained cold stress data from Min pigs. By integrating multi-omics data, we analyzed temperature adaptation in pigs and identified related structural variations. Notably, we found a 1 kb deletion upstream of the GMAP6 gene, which may be linked to heat adaptation. Lastly, we looked at how graph-based pan-genomes could boost pig breeding, focusing on genome-wide association studies and genomic selection for 6 carcass traits. We found that some genetic info can only be captured through SVs. Genomic selection accuracy showed that combining SNPs and SVs improved predictions by 2% to 4% for over 80% of traits. These results show that adding structural variations to breeding studies can enhance genome-wide association studies resolution and genomic selection accuracy. In this study, we used advanced sequencing and assembly strategies to construct the T2T genomes of 2 native Chinese pig breeds, the Min and Rongchang pigs. These genomes enabled the analysis of complex genomic regions and structural variations, revealing genetic adaptations and supporting future breeding and conservation strategies. We sincerely invite you to cite our research. If you have any questions, please feel free to contact the corresponding author.